Yeah, good afternoon, students. Welcome back to Botany second year's class. So we are doing the SAQs, isn't it? So we will continue with the SAQs from the third chapter, that is the enzymes. Because we have finished the SAQs from the first chapter, there is a transportation in plants, and the next one is the mineral nutrition. That also the SAQs we have finished yesterday. And now we will go with the SAQs from the third chapter, that is the enzymes. So from these three chapters, we can expect 2 SAQs because 4 SAQs completely from the first unit. So from the first chapter, 1 SAQ, and the second and the third, another SAQ. Otherwise, 1, 2, 3, we can get 2 SAQs, maybe from the second and the third chapter also. We cannot say that, but prepare well with the 1, 2, 3 chapters where we can get 2 SAQs. And maximum, that is 90% one VSAQ from these 3 chapters. Any one of the chapter VSAQ, we will get it. So first of all, we'll see with the first SAQ in the enzymes chapter, that is the enzyme inhibitors. Enzyme inhibitors. What are enzyme inhibitors? Enzymes are the protein molecules. Another form of protein molecules is called as the enzymes. So these enzymes are these enzyme inhibitors are the chemical substances which binds to the enzyme in such a way that they stops the action of the enzyme on the specific substrate. That is called as the inhibitors. It controls the action of the enzymes. That is the inhibition, controls, stops. Those are the enzyme inhibitors. Okay. And these enzyme inhibitors are of 3 types. That is the competitive inhibitors, non-competitive inhibitors, and the feedback inhibitors. Competitive, non-competitive, and the feedback. The first one is the competitive inhibitors. What is meant by competitive inhibitor? When the inhibitors are having the same structure according to the substance which is present for the enzymatic reaction, then these inhibitors will stop the enzyme binding to the substrate and stops the product formation. That is called as the competitive inhibitor. So during this competitive inhibitors, they are having the same structure as per the substrates. Competitive inhibitors. Competitive inhibitors means the inhibitors are having the same structure according to the substrates and they stops the enzyme binding to the substrate so that product formation is not at all seen in the reaction. That is called as the competitive inhibitors. Okay, so in this we can draw the diagram so that the explanation will be easy for us. This is the enzyme. Okay, now comes the first of all, enzyme has to combine with the substrate, and the substrate is having the like this structure. This is the substrate one, and the second one is having Okay, this is the inhibitor, that is the substrate 2. Now, what happens? This has to bind with the enzyme. The substrate 1 is having the same structure, it has to bind. But here, this substrate 2, that is the inhibitor, is having the more, it is faster than the substrate 1. So, it goes fast and attaches to the enzymatic groove. This is the enzymatic groove. At this place, it attaches itself so that we will be having the enzyme substrate complex like this. This is the enzyme now. Red one is the enzyme. Okay. Now, in this enzyme, the substrate 2 wants to fix itself. But it cannot because of a small change in its structure. So, this will be, this is the enzyme, this is the inhibitor. What it is doing? It is stopping the action of the original substrate, that is the competitive inhibitor. Okay, it is having the somewhat similar structure according to the original substrate, but it cannot fix it properly in the enzymatic groove. But it tries to fix itself. So, with that, the original substrate cannot fix in the enzymatic groove, and it will be attached to the enzyme till the life of the enzyme is going to be ended up, because time period will be there for the enzymatic actions. So, after the specific time period, the enzyme will become inactive. So, till that, this inhibitor will goes and combines with the enzyme so that the products cannot be formed. Otherwise, the product formation is delayed in the reaction. That is the competitive inhibitors. And now comes the second one, that is the non-competitive inhibitor. Non-competitive inhibitor. Non-competitive inhibitor means non-competitive inhibitors. Non-competitive inhibitors, the inhibitor does not have the same shape of the substrate, but it tries to make the product formation delay by attaching itself to the exactly opposite side of the enzymatic groove. See, this is the enzyme. Will be having the substrate. This is the substrate 1, and the inhibitor is having the different structure. Okay, than the substrate. But it will stop the rate of reaction over here. How? It will goes and combines to the enzyme opposite to the enzymatic groove. This is the enzyme. This is the substrate 1. Now, the substrate 2. So, what it does the action, the substrate 2, that is the inhibitor. Inhibitor joins the, attaches to the enzyme exactly at the opposite side of the enzymatic groove, so that in the enzymatic groove, the substrate 1 cannot fixes itself, so that the product formation cannot be seen. That is the non-competitive inhibitor. When the inhibitor does not have the same size and the shape of the substrate, but then also it delays the product formation in the reaction, otherwise in the organism. Okay, so how it will attaches itself to the opposite side of the enzymatic groove, which makes the groove widen, so that the substrate, if it comes and attaches itself to the enzyme also, it cannot fix and makes the products. That's the non-competitive inhibitor. Next comes so in this, the examples also you have to give for the competitive, non-competitive inhibitors. Competitive inhibitors example is the succinic dehydrogenase. Succinic dehydrogenase is the enzyme which is by the malonate, which closely resembles the substrate succinic. Okay, so malic acid this is the substrate 1, and the inhibitor here is a succinic acid. This is the inhibitor 2. So, which are having the same shape, but this is having somewhat different shape, so that it cannot fixes itself to in the enzymatic groove to release the products. That is the competitive. Non-competitive inhibitors, example is the metal ions, copper, mercury, silver. These are the examples of the non-competitive inhibitor. Now comes the feedback inhibitors. Feedback inhibitors means what they will do. In the feedback inhibition, a chain of reactions are going to be occurring. A to B, B to C, C to D, D to E. This is the final product. Okay. So these are the chain of enzymatic reactions are there, which are catalyzed by the different enzymes. And in this, if one of the step is going to be delayed, otherwise interrupted, then the final product formation is going to be delayed. That is the feedback inhibition. Okay. See here, the same one. A to B, B to C, C to D. And in this, 3 different enzymes are responsible for producing the final products. But in this, one enzyme is inactive. So with this, the product formation is going to be delayed in the chain of reactions. That is the feedback inhibitors. And the next one is explain different types of cofactors. Cofactors. So, what are cofactors? Cofactors are the non-protein parts of the enzymes are called as the cofactors. These are the non-protein part of the enzyme. Non-protein part of the enzyme is called as the cofactor. Okay, so the enzyme is having the protein part and the non-protein part in it. So the non-protein part of the enzyme is called as the cofactor. Okay, and these are going to be binded with the protein part to form a complete enzyme. So whenever they are binding to the protein part of the enzyme, that is called as the apoenzyme. Apoenzyme. Whenever these non-protein parts are going to be binded with the protein part of the enzyme, that is called as the apoenzyme. Okay, so these cofactors were making the enzymes which are catalytically very, very active, so that they can make the reactions faster. That is the cofactor main purpose, to make the reaction faster by the enzyme. Okay, so here these cofactors are of again 3 types. The first one is the prosthetic group. And the second one is the coenzymes, and the third one is the metal ions cofactors. Okay, so the first one is the prosthetic group. Prosthetic group means these are the lightly binded to the apoenzyme. Okay, the organic compounds, these are the organic compounds that are binded to the apoenzyme lightly, not tightly. That is called as the prosthetic group. Example in this is the heme. That is a part of the blood, hemoglobin. Okay, heme is the prosthetic group, that is which is a part of the active site of the enzyme peroxidase and the catalase. Okay, example, heme is a prosthetic group which is a part of the active site of the enzyme and which catalyzes the reaction, that is called as the heme, that is the prosthetic group. Next comes the coenzymes. Coenzymes are the okay, these are the organic compounds, but loosely binded with the apoenzyme. Okay, so these are the organic compounds that are loosely binded with the apoenzyme, and these are going to be catalyze the rate of reaction, but very less. That is the enzymatic action will not be faster with the coenzymes, but sometimes it will become the faster one. Without the coenzyme, the enzymes cannot perform the rate of reaction also. Okay, so in this, the chemical components in the coenzymes mostly are the vitamins, what we can call. Okay, so these vitamins are the most important ones in the coenzymes. The best example. So here we can say what we read with the photosynthesis and the respiration chapters, NAD plus and the NADP, isn't it? So that is the one. Both NAD and the NADP plus are contains the vitamin niacin. NAD and NADP plus both will have the vitamin niacin with them. Okay, So this is a coenzyme which is present with them, but it is loosely binded with this NAD and the NADP plus, but it is a mandatory one for going with the to show the rate of reaction in an enzyme. The next one is the metal ions. The last one in the cofactors are the metal ions. So what are the metal ions? Metal ions are the ones, the metallic ions, which requires to make the enzymatic action faster. So otherwise, without that, the enzymatic action cannot be done sometimes. Otherwise, it takes maximum time to perform to show the action of the enzymes itself. That is, those are the metal ions. So these metal ions are they will increases the activity of the enzymes by binding by binding with the enzymes on the side chains of the enzymes. Okay, so these metal ions binds to the side chains of enzymes, so that they can make the enzymatic action faster. That is the metal ions. Example, zinc. Zinc is a cofactor of the proteolytic enzyme carboxypeptidase. Okay, if you don't know this, you can go with the best example. Another one, Mg+2 is the cofactor in for the enzyme hexokinase. We know this enzyme very well because in the glycolysis, the first step, glucose to convert it into the glucose 6-phosphate, where hexokinase is the enzyme which is going to be actively participating over there. And this enzyme needs the help of the cofactor, that is the Mg+2. In the presence of Mg+2 only, hexokinase will be active to convert the glucose to glucose 6-phosphate. Without this Mg+2, the reaction cannot be faster and sometimes it may not occur also. So you can write this example also. Magnesium is also a metal. With no need for us to go with only the iron, zinc, cadmium. This also a metal. Okay. So like this, we can say the examples which we know very easier and we can easily recollect. Okay. So that is the cofactors. So cofactors means the non-protein part of the enzyme is called as the cofactor, and these cofactors are the ones which are going to be binding with the protein part called as the apoenzyme for making the apoenzyme. And these cofactors are of three types: the prosthetic group, coenzymes, and the metal ions. Prosthetic group are the these are the organic substances which tightly binds with the apoenzymes. Okay, so that is called as the prosthetic group. Example, heme, which is a prosthetic group for the active site of the peroxidase and the catalase enzyme. Okay, and the next one is the coenzymes. Coenzymes are the organic compounds which are loosely binded with the apoenzyme. These are the loosely binded with the apoenzyme, and these will be when they are present in the loosely binded position, then also they will increase the rate of reaction of the enzyme. And with the absence of these coenzymes, sometimes the rate of reaction will be delayed. Otherwise, the product formation cannot be seen. Anything can be done. Those are the coenzymes. Example, these are the chemical compounds that are present with the vitamins. So here NAD and the NADP plus, both are having the vitamin, that is the niacin with them, that is the coenzyme. Okay, next one is the metal ions. Metal ions are the ions which will be increasing the rate of reaction of the coenzymes by making the bonds on the side chains of the enzymes. Those are called as the metal ions. Example, the Mg+2 with the hexokinase. This also you can say. Okay. And the next question is the explain the mechanism of enzymatic action. Okay. Otherwise, they can ask us one more type of question, same one. Explain the enzyme substrate complex. Anything they can ask us. Okay, there is a enzyme action, otherwise the enzyme substrate complex, anything. So in this, what we have to say, how the enzyme is activating the substrate to make the products. That's the one what we have to discuss over here in the mechanism of the enzymatic actions. So whenever the enzyme is there, what the enzyme will do? Enzyme will come, attaches itself to the substrates at the enzymatic groove. Then the substrate will be released into the products and the enzyme will be having the same size. Why? Because enzyme never loses its weight or its structure by the after the end of this chemical reactions. Okay, the enzyme cannot be destroyed, nor it will not loses its weight, nor its functions, not its actions and the functions. That means enzyme will be remain same from the starting to the end of the reaction, where the chemical nature never be changed, not the quantity, not the quality of the enzyme. Nothing will be changed. But enzyme loses its life after some time. That's it. But the enzyme will not be destroyed, not it cannot be changing its structure, not its functions. That is in the reaction only what we can say. So here, what the enzyme will do? Enzyme will combine with the substrate at the enzymatic groove at the substrate specific site. Enzyme, if this is the enzyme, it is having this groove. This is called as the substrate specific site. Otherwise, the activation site of the enzyme. Otherwise, enzymatic groove also we can say. Okay. So when the enzyme, when the substrate binds to the enzyme at its substrate specific site, otherwise the enzymatic groove, then the enzyme will be highly reactive with the specific substrate and makes the products of this. And in this, the intermediate structure is going to be seen, that is the enzyme substrate complex. Okay, when enzyme combines with the substrate, that is when the substrate attaches itself to the enzymatic groove, then the intermediate compound is formed, that is the enzyme substrate complex, which is a tentative in nature. Tentative means it is a very unstable compound, unstable form, which has to go into the stable form by the formation of the products. Okay, and the enzyme will be released free from this. That is called as the enzymatic action. Okay, so in this, the catalyst is nothing but the enzyme, which increases the rate of reaction. So during this process, it never destroys itself, not it changes its behavior, not the structure. That is the enzyme. The same chemical nature will be seen from the starting till the end of the reaction. That is the enzyme specification. Okay. So here, this was the first given by Daniel Koshland. This was given by first by Daniel Koshland. Okay, so he has given the induced fit theory. Induced fit theory means a specific substrate can fix itself into the specific enzyme at the enzymatic group, otherwise in the substrate specific site. That is the induced fit theory and the lock and key theory. Lock and key theory was given by Emil Fischer. Okay. Emil Fischer has given lock and key hypothesis. So in the lock and key, what we can say, a specific substrate can fix us into the specific enzymatic group, otherwise in the activation site to make the products. That is the lock and key hypothesis. Whereas the Daniel Koshland, what he has given, there is the induced fit theory. According to this, what we can say, a specific enzyme can be fixing towards the specific substrate, but here the enzyme will not have a specific shape. It is according to the substrate, it changes its shape to hold, otherwise fixes the substrate in it to make the product. That is induced fit theory. Okay. So here what we are saying, everything we need to write and we need to conclude. The reactions, how I said, the enzyme will be fixing itself with the product and the intermediate compound. Everything we have to write in the sentences over here. And the next one, next chapter, that is the photosynthesis. In the photosynthesis, we will be having the three questions, what we can say, most important one for the supply exams. The first one is the structure of the chloroplast, and what they will give, draw a neat labeled diagram of the chloroplast. And the next question, the differences between the C3 and the C4 plants, otherwise C3 and the C4 cycles. And the next one is describe the C4 pathway. And the last question, that is the photorespiration. So we can expect in this three questions, that is structure of, that is the diagram of the chloroplast, otherwise the C3, C4 differences, otherwise the photorespiration. So when we are saying this, draw the neat labeled diagram of the chloroplast. How to draw this? If you have drawn and given the labeling part, then 4 out of 4. No need of writing a single sentence over there. But you have to draw in a proper way. Draw the disc-like structure. And it should have the 2 membranes. Okay. Now comes with the inner parts. Inner parts grana and the stroma, that is the thylakoids will be present now. So these thylakoids, you have to draw the disc-like structures first of all. Draw the oval-shaped structures so that it's easy for you to give the labeling part, and it looks like very nice also. And after this, these are going to be connected to the inner membrane and to the another grana thylakoids. Okay, and now comes the in this circular DNA will be present. Then it is having the starch granule in it. Okay. Next, it is having the stroma. Okay, now start giving the labeling part. So when we are start giving the labeling part, we will say this is the outer membrane, and this is inner membrane. Can you see this color? This is also light. Outer membrane and this is inner membrane. And these are the grana thylakoids. And these tube-like structures are the stroma, thylakoids, and this is the starch granule. And this is the circular DNA. Okay, now comes, this is the stroma. Then what we have to write, the space that is present between the outer and the inner membrane. That is the perimitochondrial membrane. Perimitochondrial space. Sorry, periplastidial space. Periplastidial space. Okay, like this. And in this, the RNA molecules will be present, as well as the lipid granules are present, lipid droplets are present. These are the lipid droplets. Okay, like this, the different, all these parts you need to give the proper labeling, and you have to give the complete picture like this. Then only you will get 4 out of 4 for this answer. Here, no need to write a single sentence, only the diagram should be neat enough and it should be having the entire labeling part. Then only you will get 4 marks for this. That is a draw a neat labeled diagram of chloroplast. Okay, so we have to move to the next question. That is differences between the C3 and the C4 cycle. When we are saying the C3 and the C4 cycle are plants, based on the question, here we have to change our answer. Based on the question, we need to change our answer. If they will be asking the C3, C4 plants or else the C3, C4 cycles. Otherwise, when they are giving the question like this, plants oblique cycles, then we have to write both the characters that are present as the differences between the C3 and C4. Or else, if only plants, then we have to specifically restrict ourselves to the plants. Usually, they will ask like this only, plants are cycles. So, in this one, the first one is the C3 and the second one is the C4. Here C3 I am writing, and here we will discuss with the C4 plants, otherwise C4 cycle. Okay, so the first one is these C3 cycles occurs, otherwise C3 plants are the temperate plants. Temperate plants, and some are tropical. Some are tropical plants, whereas the C4 plants are the tropical and the subtropical. These are the tropical and the subtropical plants. Okay, that's the first difference. Next one, in the C3 plant, in the C3 cycles and the C3 plants, what we have to say, we will go along with the stepwise differences so that it will be easy for us to know. When we said the tropical and the subtropical, otherwise the temperate plants, what could be the temperature for these plants to be tolerating? Then isn't it? That's the next topic. What we can say, the C3 plants, the optimum temperature. That means the cycle needs to occur. Otherwise, the plants can sustain the temperature between 15 to 25 degrees centigrade. Okay, 15 to 25 degrees centigrade. But here the plants can tolerate the high temperature in the tropical and the subtropical zone. The plants can sustain, otherwise can tolerate the high temperature, that is up to 45 degrees centigrade, 35 to 45 degrees centigrade. The pathway, otherwise the cycle, not the pathway, it's a cycle. The cycle can occur between 35 to 45 degrees centigrade in the C4, whereas in the C3, less temperature, 15 to 25 degrees only. Okay, and the next one. So now we have to say the points that are there in the cycles, C3 cycle. C3 cycle is also called as the Kelvin cycle. Melvin-Kelvin cycle. Whereas C4 cycle is also called as the Hatch and Slack pathway, otherwise Hatch and Slack cycle. Okay, we have to write like this: Kelvin cycle, Melvin Kelvin cycle, otherwise that is a, and the C4 is Hatch and Slack cycle. And the next one, for the in the C3 cycle, the first stable compound that is formed is the 3-carbon compound called as the phosphoglyceric acid. Phosphoglyceric acid. Otherwise, you can say it as PGA. The first stable compound that is formed in the C3 cycle is phosphoglyceric acid, a 3-carbon compound. Okay, a 3-carbon compound called as phosphoglyceric acid. That is the first stable compound in the C3 cycle. Whereas in the C4 cycle, the first stable compound is a 4-carbon compound. That is the oxaloacetic acid, OAA. And here in the C3 cycle, the enzyme which is catalyzing the first reaction is the RUB, the first compound which takes up the carbon dioxide in the C3 cycle is the RUBP, ribulose 1,5-bisphosphate. The substance which absorbs carbon dioxide in the C3 cycle, the first substance which absorbs the carbon dioxide to form the first stable compound is the ribulose 1,5-bisphosphate. Whereas in the C4 cycle, this is the pyruvic acid, phosphoenol pyruvic acid. PEP, you can call it as PEP, phosphoenol pyruvic acid, PEP. Okay, and the next one is the, for doing this C3 cycle, they need much more water. Water efficiency is more or less. In the C3 cycle, water efficiency is less, and in the C4 cycle, water efficiency is more. Okay. And in the C3 cycle, Cran's anatomy is absent. In the C4 cycle, Cran's anatomy is present. Cran's anatomy is present in the C4 cycle, whereas it is absent in the C3 cycle. Cran's anatomy is absent in the C3 cycle. Okay, and in the C3 cycle, C4 cycle won't be seen. Whereas in the C4 cycle, C3 cycle also occurs. Okay, C3 cycle also occurs in the bundle sheath cells. Next one, in this, in the C3 cycle, the photorespiration is present. In the C4, photorespiration is absent. So these are the most important differences what we can say in the C3 and the C4 cycles. So with this, we will wind up for today's session, and we will go with the other SAQs that are present in the 4th and the 5th and the 6th chapter, so that this unit will be completed. So stay tuned to the same channel for the second year supplementary exam students. Okay.